Hello, my name is Andrew Iwa, and I am board chair of the Glaucoma Research Foundation and the executive director of the Glaucoma Center of San Francisco. It is my distinct privilege to welcome you to the annual Weston Family Lecture. This annual gathering spotlights the remarkable contributions of acclaimed clinicians and scientists whose pioneering work significantly advances our understanding of glaucoma. Glaucoma, a condition that affects approximately 3 million Americans and nearly 80 million individuals worldwide, stands as the foremost cause of irreversible blindness. The Glaucoma Research Foundation's mission is to cure glaucoma and restore vision through innovative research. None of this would be possible without the philanthropic support we receive, and for that, we extend our heartfelt gratitude. Your contributions make a meaningful impact and bring us closer to a future free of glaucoma. Today's event was made possible through the remarkable support of the Weston family. Through their generosity, we have hosted this lecture for many years. While it originally began as an in-person gathering, the transition to a virtual format now enables us to welcome attendees from across the nation and around the globe. The event has provided us a unique opportunity to showcase significant developments in glaucoma research and to honor the enduring friendship of the Weston family. The launch of this lecture is attributed to Gladys and George Weston, who established it in memory of their beloved son, Danny, who tragically passed away from a brain tumor in 1998 at the age of 47. While we hold dear the memory of both George and Gladys Weston, their daughter, Jane, is dedicated to carrying on this lecture as a tribute to her parents and to honor her family's passionate commitment to the work of the Glaucoma Research Foundation. We are proudly grateful for the Weston's dedication and for our joint vision of a world free from glaucoma. It is now my honor to introduce Dr. Jane Weston, who will share a few words with us today.
Thank you, Dr. Iwa. It is now my pleasure to welcome you to the annual Weston Family Lectureship, a lectureship that was established by my parents, George and Gladys Weston, in 2007. As a patient living with glaucoma, my father learned early on not to take vision for granted, fully understanding that his sight was at risk. So grateful to be on the receiving end of state-of-the-art care, my parents sought to support the Glaucoma Research Foundation by establishing this annual lectureship, spotlighting the incredible work that is being done by experts in the field of glaucoma research. This lectureship was originally established in memory of my brother, but it is now so fitting that it honors my parents as well. Each year, the Glaucoma Research Foundation selects a speaker for this lectureship. This year, a researcher who has dedicated her career to understanding and altering the course of glaucoma. My parents always looked forward to this event, which prior to COVID took place in person. They enjoyed hosting the event and personally greeting everyone in attendance. Now, through the magic of the internet, we can share exciting insights in the field of glaucoma research with many interested people all over the world. My parents would have loved this. It is now my pleasure to welcome you to the 2023 Weston Family Lecture. I hope you find today's talk enlightening and inspirational. Thank you.
Thank you, Jane, for your continuing friendship and unwavering support. My name is Tom Bruner, and I'm President and CEO of Glaucoma Research Foundation. As Jane mentioned, the Weston family partnered with Glaucoma Research Foundation almost 20 years ago. We have immense gratitude for their collaboration and outstanding support. The memory of George and Gladys, as well as Jane Weston's dedication to continuing the tradition of the annual Weston Family Lecture in honor of her parents and her brother Danny, is something we deeply cherish. It is now my privilege to introduce our good friend and the Weston Family Lecturer, Dr. Janie Wiggs. Dr. Wiggs is the Paul Austin Chandler Professor of Ophthalmology and Vice Chair for Clinical Research in Ophthalmology at Harvard Medical School and the Massachusetts Eye and Ear. She also directs the Genetic Testing Laboratory at Mass Eye and Ear and is a co-director of the Ocular Genomics Institute. Dr. Wiggs received her PhD degree in biochemistry from the University of California at Berkeley and her medical degree from Harvard Medical School. She completed her ophthalmology residency at Massachusetts Eye and Ear and received fellowship training in glaucoma and medical genetics at Massachusetts Eye and Ear and Tufts Medical School. In addition to being an incredible physician scientist and dedicated glaucoma specialist, Janie is a scientific advisor to many organizations, including Glaucoma Research Foundation. It is my great honor to welcome Dr. Janie Wiggs.
Thank you so much, Tom. And thank you to the Weston family for this great honor to also give this lecture. So I wanted to start this lecture with a slide that provides an overview of the disease process glaucoma. And glaucoma clinically is actually a very complicated disease. The disease starts with a problem in the front of the eye causing elevated intraocular pressure. And then that elevated intraocular pressure damages critical structures in the eye that are located in the back of the eye, specifically the optic nerve, which you can see the kinds of defects that we see when we look in at the patient and that image on the top. And then the damage to the optic nerve causes severe vision loss and loss of visual field that we measure also in the clinic as a sign of disease progression. So our goal in terms of treating glaucoma is really to try to prevent the disease. Once the optic nerve has become damaged, there's nothing we can do to bring the vision back. So our whole goal is to prevent that damage from occurring. And we obviously want to try to do that at the very earliest stages of disease, when we can first treat before we have any irreversible blindness occurring as a result of the damage to the optic nerve. But the trick is trying to identify people at that very early stage before they've really had damage. The only thing oftentimes that we can rely on is elevated intraocular pressure, but many patients go on to develop optic nerve disease without ever elevating their intraocular pressure. So it presents a real screening problem for us to be able to identify people who really are at risk for disease. So the approach that we've taken in my lab, and really there's a whole field of investigators that do this kind of work, is we've taken the approach of using genetics, the genes that you carry around with you your whole lifetime, to use this genetics as a way to identify your risk of developing glaucoma. So this genetic approach relies heavily on the fact that most types of glaucoma have very strong heritability, meaning in fact that most types of glaucoma are caused by changes in your DNA and changes in genes that are encoded by the DNA. So how does it help to find these genes that cause glaucoma? Well, if we can characterize those genes and identify them, we can use those genes for genetic testing to identify people who carry mutant forms of the disease that actually increase risk of glaucoma. And then also the genes, once we understand what they do in patients' eyes, the genes can then identify potential novel therapeutic targets for gene-based therapies. And I'm going to talk to you a little bit today about each one of these, these sort of plans for using genetics and where we are today and sort of where we hope to be in the future. So I want to start just by giving you an overview of glaucoma genetics. And an important feature here is that glaucoma really affects people across the entire lifespan. There are babies that are affected by glaucoma. There's children affected by glaucoma. Young adults are affected by glaucoma. And then, of course, older adults are also affected by glaucoma. The younger kinds of glaucoma, the kinds of glaucoma that affect children and babies, these are inherited as sort of rare traits, but they have very strong heritability, very strong mutations that cause these diseases. And we can use these genes for genetic testing in families. And I'm going to show you an example of that in a minute. The adult forms of glaucoma are more common in populations, and their inheritance is also somewhat complex. But recently, we've had amazing advances in understanding the genetics of these common complex forms of the disease. And already now we're beginning to use some of that genetic information also for genetic testing that can help identify people in a population who are at increased risk of disease. So first of all, how do we use these early onset glaucoma genes for testing in families that have those kinds of diseases? Well, here's one example. The solid circle there is a female patient who came to see me in clinic a few years ago. She had been diagnosed with glaucoma at age 20 and had surgery for glaucoma. She had had a very severe disease course and had 4 children, and she was worried that as her kids were getting older and sort of approaching the age where she herself had developed glaucoma, that maybe some of them would develop glaucoma as well. So she wanted to have genetic testing done, and especially because her oldest child had been to get glasses. And at the exam, they had noted that his intraocular pressure was slightly elevated at 22 to 23. So she wanted genetic testing to be done to see if there was any gene mutation that she had that maybe she had passed on to her children. So we did genetic testing, and in fact, we found that she did have a mutation. She had a mutation in a gene called myocilin. I'm going to talk to you some more about that also later in the talk. And what we found, in fact, is that her son, with the slight elevation of pressure, did, in fact, have the same mutation as his mother. So how does this information then help us in terms of taking care of this family and the people in this family? Well, first of all, for the son who has elevated pressure, and now we know that he does have this same mutation as his mother that can lead to a severe form of disease, we might want to start treating him at an early stage. At this point, he still hasn't damaged his optic nerve, but we know that he could go on to have damage to his nerve, and therefore we really would want to start treatment now. The youngest child in this family also had the mutation. He doesn't have any abnormalities at the moment that would require treatment, but the benefit here is that we know that we need to watch him really carefully because he has this same mutation that could also lead to severe disease. And then the 2 children in the middle of the SIP, they don't have the mutation. And this is perhaps the best news that we can give this family, is that these 2, their risk of developing glaucoma is really no greater than that of the general population, and they don't need any special early treatment or disease monitoring. Now, there's a handful of genes that cause these kinds of early onset diseases in these families that we can use for this kind of testing, and they're all listed here in this table. I'm not going to go through each one of these and what they do, but I'm just raising this issue to emphasize the point that for all of these genes, we can give this kind of information to families who have mutations in these genes. A problem that we have, though, is that when we test these families with this panel of genes that we've identified so far, we only find mutations in about 20% of patients. So if we find a mutation in a family, that's great news, and we can do so much with that, and it's really wonderful for the family. But for the 80% of people that come to us for genetic testing, we can't be as helpful. So there's a great need for identifying new genes that cause these early onset forms of glaucoma. And we've been working very hard to do that in our laboratory. To facilitate this novel gene discovery, we're using state-of-the-art, cutting-edge whole genome sequencing techniques to really be able to identify every possible mutation that we can find in these families. And we're also expanding our search for genes that cause these diseases to populations other than just the regular populations that we might find in the United States or other European countries, going into more ethnic, diverse populations that have different prevalences of the disease, which could help us in our search for novel genes. So going back to our lifespan of glaucoma, I want to shift now and talk about the adult forms of glaucoma, which, remember I said, have a more complex inheritance. So what that means is that unlike the early onset diseases, where a single gene mutation that you can test for is enough to tip the balance from health to disease, the adult onset genes, each genetic defect that's associated with these conditions just makes a small contribution to disease risk. But if you have enough of these, then you can tip the balance from health to disease. So when we do genetic testing for adult onset glaucomas, it doesn't make sense to do like a single gene kind of test. Instead, we really have to test for a panel. We have to do something that's called a risk score, so that we really know, like, if there's 15 genes, how many does somebody have? So this field has advanced tremendously over the past several years. And when we do this kind of genetic testing now, we do something called a polygenic risk score. And this is a little bit complicated, but is just such an important advance. I want to spend a few minutes on it. So a polygenic risk score is really a measure of a genome-wide risk of disease. So all of the DNA that's in all of your chromosomes contributes to your risk on some level of every possible disease that you could have. Most diseases, your risk is very, very small, and you have very few risk variants. For some diseases, your risk might be higher, and you might have a lot more risk variants. So for glaucoma, what we do to calculate this genetic risk score is we use all of the genetic risk variants that we currently know of that contribute to these adult onset forms of disease. So how do we find those genetic variants? Well, we do something called a genome-wide association study. And here on this panel, this is what the results of a genome-wide association study might look like. In this kind of study, we take thousands and thousands of patients and thousands of controls, and we compare the distribution of DNA variants between cases and controls and identify those that are more likely in cases to cause disease. We then plot the results of this in this so-called Manhattan plot. And each one of these peaks represents the statistical value of association of a particular DNA variant or DNA risk factor for disease. So we have this whole collection now of many, many DNA risk factors that are associated with glaucoma. And we calculate a polygenic risk score for each individual by really simply tallying these, just adding them all up for each person. So how many does each person have? And in a population then, what you might find is that most people are going to have an average number of these risk alleles or these risk DNA variants. Some people have very, very few of them, and they have a very, very low risk of disease, while others have a great number of them. And those are the people that are likely to have the highest risk of disease. So what we've done is we've done this genome-wide association study to find all of these risk variants. We've created this polygenic risk score by adding up all the variants in different people. And then we've looked at the clinical features of those people to understand how this genetic risk based on this score is contributing to disease. So here are some of the things that we have found so far. First of all, this graph here with the blue bars shows that people who have the highest genetic burden of the gene variants just that define this polygenic risk score, these people are at least 5 times higher to have a 5 times higher risk of developing disease compared to people in the lower percentiles. So indeed, people that have the highest genetic burden of polygenic risk score are most likely to develop glaucoma compared to people who have a very low genetic burden. Now, also really importantly, is that people who have glaucoma and also have a very high genetic risk score are much more likely to develop the disease much earlier in life. They're also much more likely to have a more severe form of the disease that's characterized by more damage to the optic nerve, which is described here as a thinner RNFL, and also a higher rate of having to have incisional surgery to control intraocular pressure. So people who have a high polygenic risk score are more likely to have glaucoma, and if they have glaucoma, they're more likely to have more severe disease, characterized by earlier disease onset and more severe damage to the optic nerve. So how does this help then in terms of our screening and you know, how we can apply this kind of genetic testing to a population? Well, let's say that we have a population like this with a lot of people who don't know what their glaucoma risk is, which is most of the time, people don't know what their glaucoma risk is. Well, if we calculate a polygenic risk score for this whole population, we can identify those people, the top percentile, or the top 90th percentile of these people who have high genetic risk. We can also identify then the people with medium risk and the people with low risk. But the people with high risk, we can really target our surveillance and our careful early detection of disease to this group of patients with really high risk who really need the tightest surveillance and the earliest possible treatment for disease once it begins. We think that this is going to be something that really is going to be an advance in terms of taking care of glaucoma patients. We're doing an interesting clinical trial right now in our hospital here and also in collaboration with our colleagues at Mount Sinai Hospital in New York City, where we've calculated this polygenic risk score for patients in our communities and then identified people who are at high risk and are bringing them into the clinic for an exam to really get an idea of what kind of disease this high polygenic risk suggests or predicts. And we found some amazing information already that suggests that the polygenic risk score is really going to be helpful in terms of risk stratifying populations for people who will benefit from early treatment and surveillance. So I wanted to spend a couple minutes talking also about how we can use genes for treatment. And I'm not going to get into great detail about this because it can be kind of scientifically intense, but I wanted to give at least one example and then talk about some other genes that could potentially be targets for gene therapy. So the example that I really want to spend some time on is myocilin. And myocilin is one of the first genes that was ever found for glaucoma. It's actually one of the genes that can cause these early onset types of glaucoma in the families. We've found a number of different mutations over the years in myocilin, and these cause these dominantly inherited forms of glaucoma, abbreviated juvenile open-angle glaucoma, which is a familial kind of early onset glaucoma, and in fact is the disease that affected that family that I presented at the beginning, and in some cases also contributes to some of the adult onset cases. So what's interesting about mycilin is that it's normally secreted from the cell into the tissue around the cell. And so here on the left panel, you can see that the mycilin is being normally secreted out into these subcellular tissue structures, and then out of the cell here is these little blue dots. For the mutant mycilin, what you can see is that it's not moving forward out of the cell as readily. And in fact, we see it accumulating inside the cell. You can see that it's stuck in these structures here, stuck a little bit here, and just a few little blue dots manage to get out of the cell. So what happens when the mutant mycilin gets stuck in the cell here is that it causes damage to the cell. The cell doesn't like to have it there, and it clogs up activities and clogs up protein passages so that in fact the cell ends up dying. And as a result, then the pressure in the eye goes up because these cells are important for maintaining pressure. An interesting point is that people do just fine if they don't have mycilin. So the interesting thing is that it's only when you have a mutation in mycilin that it causes a problem. If you're missing mycilin altogether, it's really not a problem. So that gives a clue to how treatment can be developed for people who have mutations in this gene. So a few years ago, Val Sheffield at the University of Iowa did a really fascinating experiment that is an important sort of preclinical experiment for developing potential gene-based therapy for people who have mutations in mycilin. What he did is that he used a mutant mouse, so a mouse that has mutant mycilin, and this mouse also has elevated intraocular pressure and damage to the optic nerve. And he used a very modern technique called CRISPR-Cas9, and he used CRISPR to remove the mycilin from the mouse. And when he did that, what he found is that the pressure in the mouse went down. So it was a really interesting proof of principle where he took this mouse with a mutation, applied this gene editing approach to remove the mycilin, and in fact, the pressure in the mouse went down. So here on these two panels, the purple line is the line from the mouse that was treated, and the pressure is lower at 2 months in that mouse, and the blue line is the untreated mouse with higher pressure. A really important thing that he did is he showed that you could wait a few months before doing the treatment. So this is, if the disease is affecting a very young child, you could actually wait and do the treatment when the child is a little older, or even in adults before significant damage has occurred. So there are a number of other genes that cause early onset glaucomas that are also potential gene-based therapeutic targets. So I've told you a little bit about mycilin, where we have an opportunity to edit and remove the mutation. There's also an important set of genes called TEC-angiopoietin-1 that also cause early onset disease, early onset glaucoma, and can contribute to adult onset disease. Here, there's an interesting approaches to try to improve the function of these genes to be able to lower intraocular pressure. There's a couple of other angiopoietin-like proteins, or angiopoietin-2, angiopoietin-like 7, that are also genes that could be targets for treating angiopoietin-like 2 by inhibiting a microRNA, and angiopoietin-like 7 by inhibiting gene expression can also lower intraocular pressure in certain cell and animal models. And then finally, a treatment that I think is really interesting, potentially on the horizon, that we've been working a lot with primary opening of glaucoma, adult onset glaucoma, is being able to increase the availability of NAD and NADPH. These are normal metabolites that are found in the mitochondria in the cell, which are sort of the powerhouses of the cell. that provide power and keep the cells functioning properly. We have found that genes that are involved in the production of these critical metabolites can increase risk of primary open-angle glaucoma, suggesting that restoring a supply of those metabolites in those people could be very helpful. So I just want to give a quick summary and say that, genetic testing using early onset glaucoma genes can identify carriers of these important high impact mutations, allowing for surveillance and early treatment in families that have these mutations. I can't emphasize enough how important it is in these families to be able to identify people who have these mutations, and also the kinds of opportunities that we can give for genetic counseling and informed risk assessment for these families. Our genome-wide association studies have identified hundreds of these risk variants or DNA variants that influence disease risk for adult onset diseases. And we can now use these risk variants in aggregate to create these polygenic risk scores, which can define individuals at high disease risk who have more severe disease and with earlier onset, allowing us really to screen populations using the polygenic risk score to be able to identify people who need to have this careful surveillance and monitoring in order to prevent irreversible damage to their optic nerves. And then finally, there are a number of gene-based therapies that are emerging for early onset glaucoma and even some for adult onset disease. I think the more genetic work we do, the more targets we'll find that will be useful for these kinds of gene-based approaches. I wanted to just spend a minute on some of the current work we're doing and some of the future work that we have planned, because I think it's really important to understand that even though we've had just tremendous progress in the past couple of years, or past couple of decades and the last couple of years, we still have just so much work to do in this area. And I think by accomplishing this work, we really are going to be in a position where we can offer better care for patients affected with glaucoma. So we really do need to have a better understanding of the genetics of early onset glaucoma. We need to be able to improve that diagnostic yield from 20% to more, at least 50%. And so this is very dependent on new gene discovery. We only know really of just a few genes that cause these early onset diseases. So we need to put a lot of emphasis on new gene discovery. We're also working hard on understanding more about the genes and mutations that we already know about that cause these diseases, so that when we do genetic testing for these diseases, we can give people really good information about the mutations we find. There's a consortium, it's called ClinGen. It's supported by the National Human Genome Center or Genome Research Institute. And the goal of ClinGen is to annotate or characterize all of the genes and mutations that cause all of human disease. And just recently, in the last few years, we've been able to start this program for glaucoma genes, which is very exciting and will definitely help our genetic testing. Part of the work that we're doing with our whole genome sequencing effort to find new genes for early onset glaucoma is to look for these non-coding effects, so things that are outside the protein structure that may influence disease progression, and also what we call polygenic effects, even in early onset disease, things like modifier genes or other kinds of effects that can influence the severity of these early onset glaucoma genes. In terms of the adult onset disease, we do need more research, I think, on the clinical outcomes for polygenic risk scores, and I think we are really beginning to get there and get some really interesting information now about the clinical features that are associated with this high genetic burden defined by the polygenic risk score. I've shown you a couple of things already: earlier disease onset, higher intraocular pressure, and more severe optic nerve disease. But we're finding too that people with high polygenic risk are so much more likely to develop disease compared to lower risk, that we think it really will be an important tool in the clinic soon. We do need to know more about things that can modify the polygenic risk score, things that might, people that should have glaucoma based on the score and don't, or people that shouldn't have glaucoma based on the score and do. In both of those situations, we need to understand more about the factors that can modify that. And we also need to understand how polygenic risk can be passed on to other people in the family, because of course we all know of these families with multiple people affected. So with that, I'd just like to acknowledge a lot of people who really, I went over a lot of this work kind of quickly, but a lot of people who really have contributed. These are all the people that are part of our International Glaucoma Genetics Consortium, which is responsible for the genome-wide association studies that have made the development of polygenic risk scores possible. And then these are other people that we work with in my group, in our laboratory, and our funding. So thank you very much. I'm more than happy to answer questions.
Well, thank you, Janie. What a fascinating presentation, and so easy to understand. You just made it all so very clear. But we have quite a few questions from our attendees. And so let's get started with those and see where we go. The first one I have is about family history, which you really talked a lot about. And the question, and in some ways, I think you maybe have answered this one: how early should you start getting tested, and what can you do to lower your risk?
Yeah, so that's a really good question. When I tell patients who come to the clinic and say that they have a family history of glaucoma, I always ask them how old their relative was when they developed disease, because I think that's an important guide. So, if somebody developed disease, actually the example I gave, she developed disease when she was 20, and her son was approaching 20 and already had some early signs. So, if that's, I think that's a good rule of thumb, that whatever, the disease onset was in the family, that's kind of a good age to begin to look. But it isn't always perfect, and of course, I would also advocate that people should be screened. If there's a strong family history, people should be screened, even potentially at younger ages, and certainly at any point where they start complaining about problems with their vision.
Well, it's interesting because you could speculate, I guess, that people that get glaucoma at a younger age maybe would be in this 20% that have the higher polygenic risk factor. So that's a fascinating, and that's the first time I've really heard that. And we do find cases where people are identified when they're getting their college physical, for example, and then find out that it's in their family and wish they had been screened, and they already have lost some vision.
Exactly.
When considering family history, should we only consider immediate family, or what about extended family as well?
Yeah, that's also a really good question. I get asked that question a lot. I mean, it depends a lot on the type of inheritance that we're looking at. And so for things that are like, autosomal recessive, you can have distant family members who are affected and the gene is still being sort of passed on in your family. So I really think that where screening is not that difficult to do, if there's someone in your family that you know of, whether it's an immediate relative or an aunt or a cousin or something, I think it's worth doing, because there are some, most of the time it's going to be immediate family members that are most relevant, but sometimes it's people who are more distantly related. And so it makes sense to do the screening at least once.
Yeah. Now, is there available genetic testing today? I mean, is this something that someone
Yeah.
who's, for example, watching this webinar and is interested could get their risk factors evaluated and likelihood of developing more serious glaucoma or developing glaucoma at all?
Yes, there are. So first of all, a number of different companies and also a number of different labs, like our lab here, offer genetic testing for the early onset glaucoma genes, and that's pretty standard. Most of the time, insurance pays for at least a significant portion of that. One thing
Good.
I always want to remind people is that genetic testing is a one-time test. So you don't have to do it again. Once you've had the testing done, it's done. And so there's that. But what's really interesting is that the polygenic risk score testing is also emerging into the public. And so there are companies also that are these companies that will do direct to consumer genetic testing or ancestry, for example, testing. They also now will add a polygenic risk assessment for diseases like glaucoma. And I think that as we, this is pretty early days still for the polygenic risk score. It's really only been a few years that we've, even had it as a research tool. But I think that rapidly we're learning that it's clinically very relevant. And so people who are in that top, percentage of the risk alleles, I mean, they really do have greater risk of disease, and they really do have greater risk of more severe disease. And so it's a tool that I think we're going to start to use more and more often.
Well, it's good to see that is being used. I mean, you've worked for many years to develop all this information, and now it's actually in use. So congratulations.
Yeah.
Here's another interesting.
It took a while to get there. Yeah.
Well, I mean, that's one of the frustrating things about medical research is it takes time, but safety is so important, and not, misleading people. So I think caution is important. Another good question. What about injury or viruses? Can these cause, say, a gene to turn on, or can those be impact the glaucoma risk factor or the development of glaucoma?
Yeah, it's a good question. I mean, you're talking to a geneticist. I think everything is genetics, but the truth is that there are probably strong environmental effects from things like trauma and viral infection that can cause types of glaucoma that probably are not related to underlying risk. But one point I do want to make, and that is that if you have something like a virus that could, raise your pressure, which in fact there are some that do, or if you have injury to your eye that could raise pressure, which in fact it can do, if you couple that level of risk with also a high background genetic risk, then that could really tip you over into disease, where somebody else who has that same trauma to the eye but has low genetic risk may not have the same consequences.
Fascinating. Now, how is glaucoma related to gender? I noticed, interestingly, in your sample family I think there were both male and female with and without the myocilin gene. But are women or are males or females any more likely to get glaucoma? What's the data on that?
Yeah, so there have been some studies looking at, specific hormones like estrogen, which of course are in women and not men. And actually, our group has done some of this work also. And there seems that it could be, one factor that might contribute. But in general, we find pretty equal numbers of males and females affected in our various studies. So I don't think it's a huge effect in terms of women or versus men, but we probably need to do some additional work there to be able to say that very clearly.
I think this is another good question. If, let's say, you're already diagnosed, so you have glaucoma, you know you have it, and it might be good to know, are you likely to progress, for example, someone who's diagnosed? And what comes to my mind immediately is your discussion about early diagnosis or early having glaucoma early could mean you're more predisposed to having more severe glaucoma or not. And would that mean genetic testing would be important for someone who has glaucoma, or what's your thought on that?
So yeah, I do think that genetic testing is also important for people who have been diagnosed with glaucoma, because we have found in some of these studies that among glaucoma, affected individuals, people who have glaucoma and also have high genetic risk are more likely to have a more severe course. So they may be the ones who just keep progressing no matter what medication you're on. And the people who actually need to go on and have surgery. We also did a study just recently. It's still going through the peer review process. But looking at people in the ocular hypertensive treatment study, which is really one of the, kind of landmark glaucoma clinical trials. And we showed that people who have high intraocular pressure, but convert then to having damage to their optic nerve, the people who do that are more likely to have the high polygenic risk score.
Yeah, so it tends to
even in a
confirm.
setting of,
Yeah.
right. So even in a setting of high intraocular pressure, there's a lot of people with high intraocular pressure who don't develop damage to their nerve. And what we have found is that the people who are more likely to develop damage to their nerve are also the people who have a higher polygenic risk score.
Now, you did mention the CRISPR technology and the idea of treating the mycilin gene in an animal. Is there any movement to making that available for people? When do you think? I mean, I know that well, I'm sure you know well, the first gene therapy that's FDA cleared for Leber's congenital amaurosis is so there is a human gene therapy that is FDA cleared for a very rare type of blindness. What are the chances of a gene therapy for glaucoma being approved? Is that going to come anytime soon?
Well, I really hope so. I really do hope so, because I think mycilin is such a great target. For some of the reasons that I said in my talk, it's easy to just take it away. You don't need to worry about losing it, because if you lose it, it doesn't cause a disease. It only causes a disease when it's there with a mutation. So it's such a cool thing. It's a really great target for CRISPR approach. There've been a number of, and we already have this preclinical model that shows that it at least in an animal, it can work. There have been a number of industry sponsors that seem to be interested in it. They kind of come and go. I'm hoping that it will stick for one of them, because I think it would just be a really wonderful opportunity to offer some, very gene specific therapy for glaucoma for people who have mutations in this gene. And it's the most common genetic cause, single gene cause of glaucoma. So
Yeah.
I think it would impact a significant number of people.
Now there's a significant amount of data to show that certain ethnic groups seem to have a higher prevalence of glaucoma, and I wonder, have you looked at whether they have a higher polygenic risk factor or not? Do you see anything in your data to suggest that there's a possible link toward ethnicity?
So that's a great question, and it's really a current research topic. And there are a lot of people looking into this right now. One of the limitations that we have is that the genome-wide association studies that have been done so far, unfortunately, have been centered on a lot of existing population data, which comes from the US and Europe, and so not a lot from Asia, but not as much from African countries. And so we are a little lacking in data for African Americans, who are a group that seem to be preferentially at risk.
Hmm.
So we would like to have a lot more information about that. And I think it is really important. The only thing I can say so far is that, again, going back to the OATS study, we've looked at this polygenic risk score that we've created based on the existing GWAS, which does include a few Africans. And that score worked equally well in African Americans and in the Europeans, the white Europeans. So in terms of showing this increased risk of developing glaucoma in those populations. But we have a lot more work to do in
Well, but you accomplished so much, and I have to say I've followed your work with great interest, and frankly, this was the first time I really understood this whole concept of the polygenic risk factor. So I think that's very exciting. Another good question, and this does come up frequently, is are there clinical trials right now that families could join that could assist in moving this all this research forward, and might also help them to know what their risk factors are?
Yeah, well, that's a great question. So the current work that we're doing here at Mass Pioneer and at Mount Sinai is based on the biobanks, the local biobanks that we have at these institutions. So so there's a biobank here at Mass General, that we're part of. And there's about 30,000 people in it. We've calculated the polygenic risk score across all 30,000 of those people. And
Wow.
then we've identified the very top people and the very bottom people, and we're having them come into the clinic so we can examine them and see if they have disease, see what kind of disease they have, how aggressive it is, all of that kind of thing. We do like a soup to nuts clinical exam. And then the same exact protocol is being used by our colleague, Lou Pasquale, at Mount Sinai. They have a very similar biobank at Mount Sinai. He's doing the exact same thing. So that project is about halfway through. It's funded by the National Eye Institute, and it's been really exciting. We've learned a lot, and I can just say that the vast majority, if not every person that we've diagnosed with glaucoma so far, has been in the top percentile.
Oh,
So it
that's
just shows
interesting.
you how important the polygenic risk score is. Now, in terms of joining a study, we actually are going to start another clinical study here at Mount Pioneer, where every patient who comes into our clinic, we're going to offer them the opportunity to enroll in a genetic testing protocol, where we draw blood, do the genetic testing in a CLIA lab so we can give the results back to the provider and to the patient, but then follow them over time. So we also have a sense of how they're developing, what kind of features of the disease they're developing, and so forth. So that's not going to be an international study or national study yet. But I imagine it wouldn't surprise me if a lot of clinical departments of glaucoma start adopting something similar to that.
Well, I have one more question to ask, and I think I have to thank you again for all you've done, and for this great presentation and great discussion. But we'll have to keep this short. But what would you say is the most surprising thing you have learned in recent years about glaucoma? What was the most unexpected finding?
Wow, there have been a lot of things that have been so exciting to learn. But I guess one thing that has surprised me a little bit is just how well this polygenic risk score works in terms of identifying people at high risk. It really, I think it could be a game changer in the way that we, sort of approach risk stratification in populations and really trying to get treatment to people who really need it the most.
And that's always one of the biggest questions for a glaucoma doctor is how aggressive should he treat the patient? And if he could have that answer early,
That's
that
right.
might save a lot of vision. So I think that's a wonder. I agree completely with you. Well, thank you very much. This has been a
Well,
great
thank you.
presentation.
Yeah, it's been a lot of fun. Thank you very much. Yeah.
Well, thank you for your time. And thank you for your commitment to helping people affected by glaucoma. And I know this has been a long, well, almost, I guess, a lifelong effort from you. So we all appreciate your hard work. This webinar was recorded and will be available on our website very soon. If we were unable to answer your questions today, please do visit our website, www.glaucoma.org, for more information about glaucoma and our research programs on vision restoration. All our endeavors focus on our important mission to cure glaucoma and restore vision through innovative research. Part of that research includes our commitment to better understand the genomics of glaucoma and further investigate how gene therapy can be used to keep retinal ganglion cells alive and well. We are extremely grateful to our longtime friends, Mona and Ed Zander, who have supported us for over the past 22 years and recently established the Zander Family Research Fund. Their gift will support an annual research grant over the next 5 years dedicated specifically to glaucoma genetics. Since glaucoma is in their family, Mona and Ed were excited to support this important research. We are so very grateful to all our donors who continue to invest in critical research and support our patient education initiatives. Please know that every gift, no matter the size, makes a difference and brings us closer to our goal, a future free from glaucoma. Thank you and see you next time.